Let's talk about 3.2 chromosomes. Here are the understandings and applications for this topic. Let's start off with understanding 1, prokaryotic chromosomes or bacterial chromosomes. So prokaryotes have one circular DNA molecule, and this is present in the form of a chromosome. This DNA molecule is not associated with proteins, so this makes the molecule naked DNA. And since there's only one DNA molecule, the prokaryotes only have one copy of each gene. However, two copies may briefly be present when the cell replicates via binary fission. Plasmids, understanding 2. What is a plasmid? Well, a plasmid is a small extra DNA molecule, which is most commonly found in prokaryotes. They are small, circular, and naked in structure. You can see that right here. Plasmids, their presence is quite uncommon in eukaryotes. Plasmids, they contain useful genes. An example for useful gene is a gene for antibiotic resistance. They do not, however, contain the genes needed for basic life processes, so a prokaryote could survive without a plasmid. However, it cannot survive without the main bacterial DNA. Plasmids, they are not replicated at the same time or rate as chromosomes. So this gives rise to multiple copies being present in the same cell, and a plasmid may not be passed to both cells while replicating. Application 1. Cairns technique for measuring the length of DNA molecules. John Cairns produced images of DNA molecules from E. coli using this technique right here. And this technique involved, with step 1, growing cells for 2 generations in culture medium containing tritiated thymidine. Before we go on, let's talk about what thymidine is. So we already know a nucleobase of DNA is thymine, and the other nucleobases are adenine, cytosine, and guanine. But what is thymidine? Thymidine is a nucleoside. This is a nucleobase thymine, and thymidine is a nucleoside. So essentially, we have deoxyribose sugar or pentose sugar attached to a thymine nucleobase, which forms a thymidine, also notated as DT. And this thymidine is tritiated, and what does that mean? It means it has radioactive tritium. Why is it radioactive? It's because tritium is an isotope of hydrogen. And it's radioactive because this isotope is a radioactive isotope. And if you want to learn about radioactive isotopes, you should look into topic 2.1 of IB chemistry on our channel. So he grew those in these culture medium because when the E. coli replicates, we're going to have radioactively labeled DNA molecules. This moves on to step 2, where he placed cells on a dialysis membrane, and the cell walls were digested by using lysozyme, and this causes the cells to burst and release DNA onto the surface of the membrane. He then used a thin film of photographic emulsion which he applied to the surface of the membrane. He then left it in the dark for a period of 2 months. The tritium in the DNA decayed and emitted high-energy electrons which reacted with the film. After 2 months, the developed film was examined with a microscope. And the dark grains, if we look at this image right here, we can see the dark grains right here. These show the regions of this tritiated thymidine. And he produced images such as this, and this showed us that prokaryotic DNA is circular. If we go back to this image, we can see the dark grains right here. Shows the general structure of prokaryotic DNA, which is circular. We can see that there are gaps between these dark grains. That's because only thymidine or the nucleobase thymine was radioactively labeled, and we know in DNA we have 3 other bases, guanine, adenine, and cytosine, so we can infer that these gaps are filled in by those 3 other bases. But they generally show us that prokaryotic DNA is circular, and this contrasts with the linear structure of DNA in eukaryotes. How did he know that DNA was linear in eukaryotes? Well, he used autoradiography to get images of eukaryotic DNA. Understanding 3. Eukaryote chromosomes. Chromosomes in eukaryotes are composed of DNA and proteins. And the DNA in eukaryotes is long and linear. Histones, they are proteins that pack to the DNA. So these histone proteins are the proteins that are associated with DNA in eukaryotic DNA. And we can see that these yellow structures in this image are histone proteins. And the gaps between these give the appearance of a string of beads during interphase. Why an interphase? Because in interphase, specifically in S phase, chromosomes or DNA is replicating. Differences between chromosomes understanding 4. In eukaryotes, there are different types of chromosomes which exist that differ both in length and position of the centromere. Let's say that the length of some different types of chromosomes are relatively the same. How are we going to differentiate them? Well, we can look at the position of the centromere, and we have 4 different types right here: metacentric, submetacentric, and acrocentric. Essentially, the P arm, or the purple region right here, is getting shorter and shorter, and the Q arm, the blue region, is getting longer. Metacentric, submetacentric, and acrocentric chromosomes are present in humans. Telocentric, however, is not, and this is when there is no P arm at all because the centromere is at the very end. So there are at least 2 different types of chromosomes in every eukaryote, and in humans there are 23 different types of chromosomes. Every gene has a specific locus, which we discussed in topic 3.1, and each chromosome carries a specific sequence of genes. Having these genes arranged in a standard sequence along a type of chromosome allows parts of it to be swapped during meiosis, which we'll discuss in a future topic. Let's talk about homologous chromosomes, or understanding 5. What are homologous chromosomes? Well, they are 2 chromosomes that have the same sequence of genes. They are usually not identical, as some genes have different alleles. So we can see that here. So the blue chromosome right here has a capital C allele, or a dominant allele, and the grey chromosome has a lowercase c, or a recessive allele, and we'll talk about what dominant and recessive alleles mean in a future topic. But now let's talk about the second factor here. So homologous chromosomes are not connected by a centromere. So we can see over here on this image, these are homologous chromosomes right here. On the left hand side, this is not a homologous chromosome, the green part just by itself, but the green and purple parts are homologous chromosomes. You may think we have 4 chromosomes on the left hand side, but that is not true. We count the number of chromosomes by the number of centromeres. So we have 1 and 2 centromeres, so we're going to have 2 homologous chromosomes, but 4 sister chromatids. Let's compare genome sizes. Prokaryotes generally have a smaller genome than eukaryotes, and looking at this table, we can see that a prokaryote right here, a bacterium cell, a T2 phage, has 1.18 million base pairs compared to a eukaryote, such as a fruit fly, which has 140 million base pairs. The genome size of eukaryotes depends on the number of chromosomes. So as the number of chromosomes increase, of course the genome size is going to increase because chromosomes carry genes. The genome size of a eukaryote is generally correlated with the complexity of the organism, but not directly proportional. And we can see that again. Because humans are much more complex than a fruit fly, of course the genome size is going to be bigger. But when we compare humans to a woodland plant, where a plant has a much simpler complexity, the plant still has a bigger genome size than humans, so this proves the not directly proportional relationship. Diploid nuclei, or understanding 6. Diploid nuclei have 2 of each type of chromosome, meaning they have 2 full sets of chromosomes, and this is denoted by 2n. An example of this is somatic cells, and in humans we have 46 chromosomes in our somatic cells. What is a somatic cell? A somatic cell is essentially a body cell, and this is given by the many examples right here. A neuron, a cardiac cell, or a hepatocytes. They are all examples of somatic cells, and they have 2 copies of every gene except of genes in 6 chromosomes. You can see in a karyogram right here, right, diploid nuclei, we have 2 of each type of chromosome. So chromosome 1, we have 2 of each type. Chromosome 2, we have 2. Chromosome 3, we have 2. So this is an example of diploid nuclei when we look at a karyogram. Haploid nuclei. Haploid nuclei have 1 of each type of chromosome, so they have 1 full set of chromosomes, and we denote that by n. These haplonuclei are present in gamete cells, and in humans we have 23 chromosomes in our gamete cells. So haplonuclei only have 1 copy of every gene. Example of gamete cell in the human is of course a sperm cell. And we can see a karyogram for human sperm and a karyogram for an unfertilized egg cell, which are both gamete cells. And we can see that they only have 23 chromosomes. Let's talk about chromosome numbers, or understanding 8. Organisms with a different number of chromosomes are unlikely to be able to interbreed. So of course, if you're going to take a chicken which has 39 pairs of chromosomes and a human which has 23 pairs of chromosomes, of course they're not going to be able to interbreed. The number of chromosomes can change during evolution of species. So as a species evolving, the number of chromosomes are going to change, and they're going to decrease if the chromosomes fuse or come together, and they're going to increase if the chromosomes split. Comparing chromosome numbers. Application 3. Some eukaryotes have a few large chromosomes, and some eukaryotes have many small chromosomes. But all eukaryotes, what we have in common is that all eukaryotes have at least 2 types of chromosomes. So the diploid chromosome number is at least 4, because if we remember from a previous slide, if we go back here, diploid nuclei, the 2 full sets of chromosomes, it's given by 2n. And now if we look at this, all eukaryotes have at least 2 types of chromosomes. So if we go back to n in this case equals 2. So 2 times 2. And that, of course, is going to give us 4. So this proves that diploid chromosome number is at least 4 in all eukaryotes. Let's talk about karyograms, understanding 9. Chromosomes of an organism are visible in mitosis. And why are they visible in mitosis? Because in prophase, chromosomes are going to condense. The chromatin fibers are going to condense to form chromosomes. And metaphase gives us the clearest view, because in metaphase they're all lining up in the middle. A karyogram is a way to depict the chromosomes of an organism. And how does it depict that? Well, they're arranged in homologous pairs for us to see. They're arranged from largest to smallest in size. We use stains because some homologous pairs are going to be the same size. So we use stains to make the chromosomes show up. And it gives each chromosome type a distinctive banding pattern. A micrograph is then taken and digitally arranged according to size and structure. So if we're going to have homologous pairs that are similar in size, the position of the centromere and banding patterns allow us to differentiate the chromosomes that may be of similar size. Sex determination, understanding 10. In humans, we have 2 chromosomes that determine sex, and these are known as heterosomes. The first sex chromosome is our X chromosome. It's a relatively large chromosome, and the centromere is near the middle. It has genes essential for both genders, so all humans have to have at least one X chromosome. The Y chromosome is relatively small, and the centromere is near the end. It has all the genes required for a fetus to develop into a male, especially in particular the gene SRY, or also called gene TDF, which codes for sex-determining region Y protein, or also called the testes determining factor protein. And this causes the development of testes and testosterone. All other chromosomes in the body are autosomes, as they do not determine sex. Females are going to have 2 X chromosomes, and they're going to pass 1 X chromosome onto their offspring. Males have 1 X and 1 Y chromosome, and the chromosome passed into their offspring depends on the sperm cell that fertilizes the egg, because when sperm is formed, half of them have an X chromosome, and the other half are going to have a Y chromosome. And daughters are going to inherit the X chromosome, because females have 2 X chromosomes, and sons are going to inherit the Y. And we can see that right here. So a female is going to have 2 X chromosomes, and a male is going to have an X and Y chromosome. You can also see that right here. So if I get my pen, we're going to have a female right here. And this one right here is going to be a male. This is an example of a karyogram. So when we're looking at this karyogram, we can analyze some stuff. So the first thing we can see from this karyogram is the sex of the individual. Since they have 1 X and 1 Y chromosome, it's going to be a male. We'll discuss karyograms in much more detail later on. A karyotype and Down syndrome, application 4. So what is a karyotype? A karyotype is a property of an organism, which is the number and type of chromosomes it has in its nuclei. And we study the karyotype of an organism by looking at a karyogram, which we discussed earlier. There are two ways which we use a karyotype. So the first way is to determine the sex of an individual, which we did right here. We just determined the sex of this individual by looking at their karyogram. We can also diagnose chromosome abnormalities, and we do this by using fetal cells taken from the uterus during pregnancy. And an example of a chromosome abnormality is having 3 copies of an autosome instead of 2, and this is something we call a trisomy disorder. So if we go back here, we can see that every chromosome type has 2 copies, except our sex chromosomes, of course. So this individual does not have any chromosomal abnormalities in reference to trisomy disorders. Now let's talk about Down syndrome, also called trisomy 21. And this is caused by the presence of 3 copies of chromosome 21 instead of 2. But we can see that right here. So if we go to chromosome 21, we have 3 copies. We're actually only meant to have 2. So this individual is in fact going to have trisomy 21, or also called Down syndrome. And common characteristics between Down syndrome patients are that they have hearing loss, heart and vision disorders, and mental and growth retardation. An important thing to know about karyograms is how we notate them. So we always start off with the sex. And since they have one X and one Y, this is going to be a male individual. And then we look for any chromosomal abnormalities. In IB, we tend to only look for trisomy disorders. So we can see that this individual has trisomy 21. So we're going to say male with trisomy 21. Then in brackets, we put the number of chromosomes they have. And we can see since they have trisomy 21, they're going to have an extra copy of chromosome 21. So we know humans have 46. So 46 plus 1 is going to be 47, comma, and then we put the sex in, and we notate that with the chromosomes. So we have a male, so it's going to be X and Y. Now, pause the video, and I want you to have a go at notating those 2 karyograms with the same way we notated it right here. So pause the video and have a go. Okay, now let's notate this karyogram right here. Straight away, we can see that I have one X and one Y. So this is going to be a male. Now we'll look for chromosome abnormalities or any trisomy disorders. And by examining this karyogram, we can see that there are no trisomy disorders. So this is going to just be a normal male. And now we're going to put the number of chromosomes in brackets, which is going to be 46 because there is no trisomy disorders, and the sex in chromosome notation, which is just X and Y because this is a male. Yep. Now let's look at karyogram number 2. Straight away, let's look at the sex, and this is a female because there are 2 X chromosomes. Now let's look for any trisomy disorders, and we can see that this individual has 3 copies of chromosome 13. So they're going to have female with trisomy 13. And now let's notate the number of chromosomes in the brackets, which is going to be 47, because it is just 46 plus 1, because they have an extra chromosome right here, and that's going to give us 47 chromosomes. And now we just notate the sex in chromosome notation, which we're going to have just X and X. Close the bracket. That's done. So that's the end of this topic, but let's discuss this. Since they have trisomy 13, this is something called Patau syndrome, which we'll discuss in future topics, but this concludes the end of 3.2 chromosomes.